സെൽ ആൻഡ് മോളിക്കുലർ ബയോളജിയിലെ ചോദ്യങ്ങളാണ് ഈ വീഡിയോയിൽ ഡിസ്കസ് ചെയ്യുന്നത്. ഒന്നാമത്തെ ചോദ്യം നോക്കാം. ദി പ്ലാസ്മ മെംബ്രെയിൻ ഈസ് മെയിൻലി കമ്പോസ്ഡ് ഓഫ് ലിപ്പിഡ്സ് ആൻഡ് പ്രോട്ടീൻസ്. The powerhouse of the cell is mitochondria. Ribosomes are mainly involved in protein synthesis. 70S ribosomes occurs in prokaryotes. The fluid mosaic model was proposed by Singer and Nicholson. Cytoskeleton is mainly composed of microtubules and microfilaments. Microtubules are made up of tubulins. Which organelle contains hydrolytic enzyme? Lysosome. Cristae are associated with mitochondria. The site of photosynthesis is chloroplast. Cell wall of fungi is mainly composed of chitin. The largest organelle in plant cell is vacuole. Which organelle is called suicidal bag? Lysosome. Centrosome is absent in plant cell. Glyoxysomes are common in germinating seeds. Unit membrane concept was proposed by Robertson. Cholesterol in plasma membrane helps in membrane fluidity. Passive transport occurs along concentration gradient. Endocytosis is bulk transport into cell. Osmosis is movement of water. Chromosomes were first observed by Fleming. Nucleosome model was proposed by Korenberg. Histone proteins are rich in basic amino acids. Telomerase protects chromosome ends. NOR refers to nucleolar organizer region. Centromere is important for chromosome movement. Kinetochore is attached to spindle fibers. Loss of chromosome segment is deletion. Down syndrome is due to trisomy 21. Turner syndrome has chromosome number 45XO. Klinefelter syndrome is XXY. Polyploidy is common in plants. Crossing over occurs during packaging. Synaptinomal complex forms during zygoting. Chiasmata become visible during diplotin. Equational division occurs in meiosis 2. Reduction division occurs in meiosis 1. Crossing over increases genetic variation. Totipotency was demonstrated by Haberland. Differentiation involves gene regulation. DNA replication is semi-conservative. Michelson and Stark used heavy nitrogen. Okazaki fragments are formed in lagging strand. DNA polymerase synthesis DNA in direction. DNA polymerase synthesizes DNA in direction 5' through to 3'. Enzyme unwinding DNA. Enzyme unwinding DNA is helicase. DNA ligase joins. DNA ligase joins Okazaki fragments. RNA primer is synthesized by primase. One gene, one enzyme hypothesis was proposed by Beadle and Tatum. Genetic code is universal. AUG code for methionine. Stop codons are UAA, UAG, UGA. Operon concept was proposed by Jacob and Monod. Lac operon is an example of negative regulation. Restriction enzymes are molecular scissors. Echo RI recognizes palindromic sequences. Plasmids are commonly found in bacteria. Southern blotting detects DNA. Northern blotting detects RNA. Western blotting detects protein. Autoradiography uses radioactive isotopes. DNA fingerprinting was developed by Alec Jeffrey. VNTR stands for variable number tandem repeats. Sanger model is also called chain termination method. Dinucleotide lags. Dideoxynucleotides lack 3'OH group. Human Genome Project was completed in 2003. PCR was developed by Mullis. PCR requires all of this. That is, PCR requires DNA polymerase, primers, DNTPs. So the answer is option D, all of this. Taq polymerase is isolated from Thermus aquaticus. Annealing in PCR refers to primer binding. FISH stands for fluorescent in situ hybridization. So ഇത്രയുമാണ് സെൽ ബയോളജി ആൻഡ് മോളിക്കുലർ ബയോളജി എന്ന് പറയുന്ന ടോപ്പിക്കിൽ നമ്മൾ ആഡ് ചെയ്തിട്ടുള്ള ചോദ്യങ്ങൾ. ഇത്രയും ക്വസ്റ്റ്യൻസ് നിങ്ങൾക്ക് അറ്റൻഡ് ചെയ്യാൻ പറ്റുന്നുണ്ടോ, ആൻസർ ചെയ്യാൻ പറ്റുന്നുണ്ടോ എന്നുള്ള കാര്യങ്ങൾ ഒന്ന് നോട്ട് ചെയ്യാം. ഓക്കെ, അറിയാത്ത ചോദ്യങ്ങൾ പഠിച്ചെടുക്കാനായിട്ട് ശ്രദ്ധിക്കാം. താങ്ക് യു.